Hey everyone, I hope you are doing fine. We are with a very lovely friend, Abhinav, and Abhinav will talk about his research and some thoughts about machine learning. But before that, could we know a bit more about you, Abhinav, like your background?
No, of course you can. Hi everyone. My name is Abhinav. I'm from India mainly, and I'm currently finally a PhD student at Karolinska Institutet in the Department of Medical Epidemiology and Biostatistics. So I have a background in bioengineering. I did my bachelor's in biological sciences and bioengineering from Indian Institute of Technology, Kanpur, which is in India, in my region. And then I went on to do my master's in health informatics at Karolinska Institutet and Stockholm University. It's a joint program. And now, and immediately after my master's studies, I went on to do the PhD in computational pathology, which we'll more talk about in later, I think. Yeah.
Maybe actually we can directly start with what is epidemiology and pathology. I think it might be super unfamiliar words for people.
So epidemiology, I mean, I'll be probably wrong, but in very layman terms, it's like the study of a disease in a large population size. So it's like how the disease or what's the impact of a treatment on a disease, how the disease is spreading, how is it impacting the people, and it's mainly like a large cohort studies to understand to have a more macroscopic understanding of the disease variations and the treatment planning and the clinical and also like drug discovery or the how the drug is impacting a particular disease type, like for example the clinical trials or there are multiple prospective trials, etc. So in general it's like a macroscopic study of a disease in a huge population or a set of population or and if it's a big study then it can even be the worldwide population as well. And computational pathology is mainly it's the computationally or the study of the pathology slides, which is mainly the pathology is basically the study of tissues or the tissue sections or the tissue specimen for different organs or anything, and you analyze for or the blood even, including the blood, which is like basically the laboratory study of like how the different tissue or or the different blood types or the blood samples like work and in different kind of conditions, which is like if they are diseased, or it can be the tumor tissue, it can also be like a burn, etc., etc. So it's a broad field, and computational is like developing algorithms or making methodology to understand or to analyze these pathology images digitally.
Maybe we can put one of actually the slide picture like on the video. So I want to go one step back and I want to maybe talk about your cultural transition from like an Indian way of teaching to the Swedish way of teaching. Was it like easy for you to adapt, or how was this transition from bachelor to master?
No, it was not difficult, but it is definitely a huge. There is a lot of cultural differences. I mean, I'm not sure if you're familiar, but the Indian style is very traditional style where you have a hierarchical system. You have a like you're a student and then you have a teacher. You always respect it's one of the core values. Like you always respect your teachers, and also you can never call a teacher by its name. So when the first time I came here for my master's, I remember we had a professor from Greece, and his name was Vasilis. But I was trying to say his last name, and you know the Greek last names, which are very, very, very tough to say, especially for me. I mean, it was my first time not in Europe, but first time in like abroad properly, and I was always trying to say Professor, his last name, I don't remember it anymore, and it was super wrong, and he was like, who are you talking to? And then he said like, oh, call me Vasilis. I was like, it was so hard for me even after 1,000 times. He was like, he was so annoyed with me. He said, if you don't call me Vasilis, I will not answer your question.
After a few years, you are just saying, buddy, how are you, buddy? What's
Yeah,
up?
I mean, now, like for example, I know your PI as well. I call him Ardench, and it's super friendly, but which is not normal. If you go back to India and in the same environment, you have to be like professor, last name, or yeah, you have to call him professor, or I don't know if you can call sir or the sir or something like this. You can never say the first name for sure.
In the US, I remember we were calling like a doctor.
Yeah, doctor. Oh yeah,
doctor and the surname.
It depends on design. If it's an associate professor, then you call him doctor or something. And if it's a professor, then it's like
he had both PhD and MD, and also the professor. I think he just for people to call him like this.
That's nice, actually.
But it's very interesting actually, because it's quite similar to the Turkish culture. We also have this hierarchy and calling people with respect.
Yeah.
But also, I know that you also have quite like a theoretical education, and like quite detailed. And I know from like a Turkish people studying master here, like they are super comfortable after being tortured for a few years in a bachelor in Turkey. Like they find you in a very easy education system here. Did you feel the same?
Yeah, exactly. It was same. I mean, I don't know if like at Karolinska especially, like you have one course at a time. But I studied engineering back in India, and we had like 5 or 6 courses at a time, and all of them are like, I mean, you have to study simultaneously all these courses. But here it's like for 2 weeks or 1 month, you have only 1 course or 2 courses, and that too like super, super easy going. You focus mostly on, I mean, it's more, I don't know how to say, more practical. At least my master's. I mean, if you talk about health informatics, it's more practical, like looking at the broader picture of healthcare systems, how the e-health or electronic health, how the digital health is coming up, and blah blah blah. So you look at these aspects slowly, one at a time, rather than and in a more easier environment. And they don't even have exams, which is like, final exam you have to pass. Here it's either pass or fail. Which is also very, very different. I mean, my master's, we had letter grading,
because of the SU part,
because of Stockholm University. But still, it's like, I remember talking to Vasilis, the Greek professor. It was about the grade actually, like how the grading system works. I mean, typical Indian style, because I don't care about what the course is, I care about the grade. No, but I was a bit confused, like how the assignment grading happens, because it was like a whole report, and I don't know how will you judge a subjective report. So I was like, so he was like, don't care about the grade. Like
literally, gonna be fine.
everyone will be fine or something. Like, if you submit a report, it's fine. No one cares. So much.
Okay, let's go to move to the research part. So actually, what you are doing as a PhD student, or you can mention like overview of your papers, maybe. What you are interested in nowadays.
So mainly I'm working in cancer histopathology images. Now, so histopathology images are basically a general overview of a cancer patient is you will screen the patient, then you find the tumor, then you remove the tumor, you do a biopsy, which is like taking a needle from the tumor specimen inside the body, which is invasive. But then you do a surgery to remove the tissue sample. So you have a whole tumor out, and then this tumor goes to the pathology labs for further analysis to see how are the cells, how aggressive the tumor is, and like TNM staging, like what stage the cancer is, which is like how much it has spread, how big is the tumor, or how aggressive the tumor cells are. So I mainly work on breast cancer histopathology, which is the breast tumor, the tissue sections, which are like from a tumor, a section cut from the tumor, and then they analyze under a microscope, and then they analyze for different biomarkers, or it can be different types of protein, blah blah. But most important idea is to look at the grading, which is how aggressive the tumor is. If it's grade 1, then it's like it's not spreading so much, it's not like very aggressive, and then you don't have to give a chemotherapy, for example. And if it's grade 3, then it's super aggressive, then most likely a chemotherapy will be given to. I mean, it depends on other biomarkers.
It's such a broad thing, right? Now almost every country does it the same way, like quite traditional way as well.
Yeah, it's a very standard system. It's called Nottingham histological grading, which is like looking at this tumor, like surgical tumor specimens, like which is like surgically remove tumors, and then you section it, and pathologists look into the slides. But now this whole paradigm is shifting because of digitization of the microscopy, especially. Now you can scan these slides and, like, for example, we looked in your facilities as well. Like, you can do a live imaging. You can look at the sample in a live on a screen. So instead of like manually assessing these tumor samples under a microscope, now the pathologists are sitting at home actually, and then they open the slides. They put the slides in the microscope, of course, but then they look on the screen, and then they are like manually like either annotating or analyzing.
And how many doctor generally needs to annotate before like giving something final decision?
So it depends. So mostly, so if you're looking at the grading of the tumor, then there are 3 subcomponents. You look at the mitotic count, like how the cells are dividing, how much they are dividing, and then you look at the broader structure, which is like tubular formation. If it's whether the normal tissue histology or normal morphology of the tissue is maintained, then it's like a normal tissue. It's not spreading so much. But if the cancer has spread, so the morphology is distorted. It's not the same. That's basically the grading. So if it's there's a more macro structure like tubular formation, which is like if the cells have spread, then there are no tubules formed in the tissue, or then there is a microscopy like the mitotic count, and there you also look at nuclear pleomorphism, which is like how the nuclei is of a normal cell. Because the cancer cells are basic normal cells, but dividing, but going through multiple mutations and the changes and multiplying infinitely, kind of.
Even there are like a very objective ticks that you can like go through these different stages, like 3 different things.
Yeah.
But still, can it be like a bit subjective at some certain stations?
Yeah, exactly. This assessment is manual. Like pathologists looking on the screen now and manually counting, and then they have a score, and then they make it into a discrete numbers like 1, 2, 3 mitotic count, 1, 2, 3 nuclear pleomorphism, or 1, 2, 3 tubule. So and it is fine if you look at 1 and 3, but then there is so much of variability that people have found between grade 1 and grade 2, or grade 2 and grade 3, especially the boundary cases, because it's not exactly discrete morphology. It's like a spectrum. But they try to. But for the treatment planning, you cannot be like, oh, it's a spectrum. So this patient can be or cannot be given this chemotherapy or any. So that's why they make it discrete, and they see the.
And here it comes in.
And here we comes in. But there is so much of variability. Like so many patients are overtreated. I think they're mostly overtreated, and some are undertreated. For example, especially between grade 1 and 2, you can see the grade 1 patients are given grade 2, and the grade 2 patients are given mostly grade 3. But 50% of the patients are given grade 2, intermediate grade. Then you don't really know what treatment to give to this intermediate grade patients.
And also, this is from one sample, right? At the end.
Yeah.
When they get the autopsy, you have one autopsy.
Yeah, exactly.
So from one slide, you want to guess. Because there might be other parts in the body. Maybe you couldn't find the tumor right away. You're not in the perfect location.
Yeah.
Maybe you think that this is actually maybe not important tumor.
Yeah.
But maybe you got it by chance from the neighborby tissues or something.
I think it's mostly the screening problem. In screening, I think one of the main tasks is to find the primary tumor, like which is origin is also because in some cases, some cancer types, you don't even know the origin. It might be the metastatic tumor you are seeing, but it might be, for example, a tumor in liver might be due to the person had a breast cancer before. But it spread. It was in the lymphatic system, so it made a tumor in liver before you could see the tumor in breast, for example. So I think it's more screening problem, but I agree with you. But for grading, especially, they figured out this is the tumor. They take out the tumor, and then they do multiple sections. Actually, they don't look at one. They see which of these sections show the highest number of cancer cells. Or I mean, I'm not a pathologist, so I'm not an expert in this, but I know they have multiple sections stained with HNE. Then they have like you can call it a primary section where they grade and give the grading to the patient. Then they go to this tumor board, or it's called, I think, molecular tumor board, where oncologists, pathologists, and other, if you need a genotype or a molecular profiling, then an geneticist can be there to decide the best treatment plan for the patient. It's literally like a roundtable conference.
Yeah, I mean, it's very important decision at that, right?
Yeah.
And how you then come into the place now?
So since I tell you it's more digital pathology, so every digital pathology, or they are scanned on this, so you assess them on the screen, right? So now you have digital images for each of these sections. So now what we have is, like in Stockholm or in the biobank, you have multiple tumor samples from different patients. So that's where we come in. Like, we have a huge cohort retrospectively collected, not like not yet prospectively, but I mean, in few studies there is prospective. Like, prospective means like collecting as we go on. Like, new patient comes in, it's part, and then you have the tissue sample, and then you ask the patient, okay, can we include this in part of research? And then you keep on developing the cohort. Prospective. But retrospective means you had patient from 2012 to 2019, you set the defined date, and then you go back in the biobank. You get the samples from there, and then you scan them, because it's a new phenomenon. It's now they are changing. Now they are storing the primary sections as in the computers. But previously they are still the glass.
But nice that they kept them. So,
yeah, the glass slides are always kept, kind of, because back then they are also like studying it retrospectively for various, for example, for molecular profiling. Or molecular profiling is like looking at the gene expression in different tissue samples. So we have this cohort retrospectively collected from different hospital or different sites, and we have the grade label given at that time to the patient, basically.
Do you guys see any differences?
Yeah, actually,
there's a huge difference, right?
There's a huge,
not just only the way you do, but also the education of the doctors and stuff.
Exactly. So also the experience of the pathologist, and the how good the lab is. Also, or I have to mention this, like digital pathology or the pathologist analyzing on screen is not well established. It's well established in Nordics, and it's established. And I think in Netherlands it's the most established. I think they have like almost all the pathology labs are digital. No one like looks at the glass slides manually under a microscope. They look at the on the screen.
But do you see so many off when you retrospectively digital them, and then you see that actually previously they were scored, let's say 2, but actually it's 3, or vice versa?
Yeah, exactly. So there is a huge variations. There was a study also by one of the pathologists here who looked at the different biomarkers assessment in different labs and within the lab. So in grading, we know if you ask a pathologist to grade the slide, and he would say tissue section, he would say, oh, it's grade 1. And if you ask him a week after, he might say grade 2. This is very highly, so this is inter-observer or intra-observer also, like. But there is a variation in different labs as well.
Okay.
So because after a period of time, you would expect the grade 2, number of grade 2 patients, the percentage of grade 2, grade 1, grade 3 to be almost proportional if they are, I mean, in a huge sample size, if they are doing everything in a very standard way, standard manner, or following the protocols. But now there is a huge variability.
So is there a way to eliminate this subjectivity?
I mean, that's where we come in, actually. So what we do is we have these images of the tissue section, cancer tumor sections, and then we use computer vision, which is a subsidiary of computer science, which mainly deals with the images or the videos, or it's based on how we actually look at images, kind of following. And then we use these predictive algorithms. I don't know if you heard of machine learning or AI, they call it now.
Yeah.
Or
First time hearing.
Oh, really? You got a nice sense of it. Now that we use these deep learning based models, which are quite prominent, they are, I mean, you must have heard of it somewhere, like the convolutional neural network based algorithms to learn the histomorphology from these images and see if we can predict the right grade, so that we have more reproducible, and so we can assess the pathologist in assigning, for example, if pathologist is assessing, we can use the model or the algorithm and to see, and then they can recommend the pathologist, oh, it looks like a grade 1 or grade 3 or grade 2. But it's a very hard problem though.
I think one thing I could tell is because of the standardization, because everyone does it differently in different lab, different practices, different instruments, I don't know. That's why you have a different sets of data, which you cannot maybe normalize it easily.
Also, I think as you said, also the staining protocols, or how long the slide was stained and left before analyzing. So because H&E is one of the standard, hematoxylin and eosin stain is one of the standard stains, and if you keep it for a while, then you can see it starts getting a bit diluted.
Okay.
So this can change how even for the pathologists it can make a problem actually.
But can you have a model to able to choose only the slides that are reliable? If it is certain, like out of parameters about the labeling, digitalizing, etc., actually just exclude the data. Like another model just to pick up the sample.
But think of it this way: you have a new cancer patient, and you basically need a model which can adapt these variabilities rather than reject it.
Otherwise, you need to do another autopsy, right?
But if the tumor is small, you have only one. I mean, there are multiple limitations to it. I mean, you cannot ask a pathologist, oh, this section looks really bad. Can you stain it, or can you do it again or something? I mean, now these protocols are quite standard, but it's just like the staining. I mean, I'm only talking about color variability in the images, but there are multiple scanning.
I mean, brightness. I mean, there are so many things.
If you use a different company of the scanner, then also it's different. So it's.
What is the problem now? How far we are? Like, can we now actually? Are they using it already? Like,
in few cases, yes and no, because so I told you about the problem of assigning mostly grade 2 to all the patients, like 50% of the cases, where we in a subgroup.
So these are false positives.
Not false positives. It's hard to determine which treatment to give. So with one of these models, you can actually you can divide these patients into low and high risk categories, the intermediate grade into.
Because this is also something I don't understand. If it is that tricky, also like important being 2 and 3, why they don't enlarge the scores a bit more, like 1 to 5? And if it is something like 2, 3, then doesn't matter actually whether it's 2 and 3.
I mean,
like make it a bit more like a dynamic spectrum instead of like a narrowing it, like even making more drastic.
Yeah, but ideally you need a binary rather than. So what you said is like, okay, yes, looking at it as an ordinal ordinal is like 1, 2, 3, 4, 5, 6 in an order. So that's why it's called ordinal kind of ordinal regression problem as well. Like instead of looking at 1, 2, and 3, you look at it as like 0, 1, 2, 2, 9. I mean, or 1 to 9 at least. But then in the end, for the patient, you need a binary. Either it's a low risk group or high. You cannot have like.
So even in the high risk group, there is only just one way of treatment. So there are not, for instance, if it was 1 to 9, and above 5 was like an imported patient.
Exactly, yeah.
So there are not different treatments for 5, 6, 7, or that cannot be.
No, exactly. We don't have. We are limited with the treatment options as well. I mean, also you cannot use. Also, grading is done in a very subgroup. Like if it's a triple negative, which is like, I don't know, ER negative, estrogen receptor negative, ER negative, which is a very highly aggressive. You don't see anything. You have to give chemotherapy as of now, because we don't know how to deal with this such aggressive type of cancers.
But actually, that kind of innovations might change the way you label, because also there's a huge improvement in the treatment of cancer, right?
like hormonal therapy,
Exactly,
immunotherapy,
exactly.
and radiotherapy.
So now you don't just do chemotherapy, right?
Yeah.
So in the future it might be actually different scales.
Yeah.
But then you need to change the model. But still you have the digital versions at least.
Yeah, I mean, depends on the drug discovery. You can also even see the impact of the treatment. Not only looking at these biomarker prediction, you can also look if you have given this treatment or chemotherapy, or in this subcategory, which is like which is biomarker positive or hormonal marker positive or negative. If whether the drug is working or not in this subcategory also. Because right now traditionally we are giving chemotherapy to all the patients, but suddenly then we realize, oh, we are over treating so many patients because the drawbacks are then with the advancement of molecular profiling, especially gene expression, then you come up with more like hormonal therapy, then more precise treatment.
Yeah, precision medicine.
Precision medicine, exactly. And then we are going further in we are in the parallel of the precision medicine. We are like, from these images you can learn the histomorphological or histomorphology of the cancer in order to separate the patient into more specific treatment rather than.
I have 2 questions.
And now, interestingly, you can also, in some cases, you can also even predict gene expressions only from these images.
Yeah, these are ridiculous. I mean, I've seen some papers, like even without any labeling and so, just from the 2 sheet stuff, like a bright field.
Yeah.
People somehow correlate with them some expressions of RNA and so.
Exactly.
I mean, it's mind-blowing.
Yeah.
So one question is, do you also do follow-up studies in a way that, let's say you label the person as a tool and treatment is not necessary, let's say, but it turned out actually it was more serious case, and do you use such information and then actually you put this image as a more importance even if it was not labeled previously or such?
So what we have is like, I'll tell you about the previous study. So my study was also very similar actually. So you have a grade 2 case, like intermediate case, and then you don't know what treatment to give to this patient. And you use the model to divide it into low and high. Right. And there was this paper from a group. So you train a grade 1 versus 3 model so you train a model which can differentiate between grade 1 and 3. And since you assume there is a more continuum, so you can see, okay, grade 2, you can differentiate grade 2 into more grade 1 like or more grade 3 like. And then you can make it into a category of low and high risk.
That's very cool.
So that you don't have this problem of under over, then you have like, okay, and then you look at the risk groups, low and high, if it's more associated with the survival or not survival or something like this. Of course, you have to look at the treatment effect as well.
And another question, actually, is very interesting. So at one point in our medicine, we want to go to the precision medicine, so personalized medicine. We want to take every case individual and treat accordingly. But at some point, we invest a lot to the machine learning. And at the end, these machine learnings are using a lot of data to have a holistic understanding and generalizing the stuff, right? At the end, you generalize and predict. So how actually these are contradict to each other? Actually, they are in the same harmony, or is actually they're totally different?
I think they are in the same harmony because my group or our group, shout out to Matthias. No, we are called predictive medicine group also, and we also focus on precision medicine. So what you do is machine learning.
But your model is based on thousands of data and not from this person.
So precision medicine is not about individual. You have to find the subcategories of each patient. So, for example, if this gene expression positive or negative, and you have a treatment for this particular gene, so it's not like one patient. You're trying to make a more specific treatment for a smaller group. You're creating small groups. I mean, it's the same thing what you said, but individual is like a bit more misleading. You can never have a particular marker for one patient or something like this. It's like a subcategory of the patient which will have more specific treatment, and they will get more benefit from it rather than be having a general treatment which is given to everyone.
Even in subpopulation of group, we have actually quite many
subcategories.
Yeah, like also data as like a number of people.
So
enough that statistically meaningful.
So in predictive or in precision medicine, how machine learning is used in big data is to find these subcategories. So if you have a huge amount of data and good amount of predictors or variables or biomarkers, then you're assuming like you can differentiate these subcategories for a completely new patient, which is like you're predicting on the new set. So that's where like the statistical learning or the machine learning comes in. And machine learning is basically the extension of statistical learning, which is like finding association between different variables. And then if you can project this learned association on a completely new set.
And how do you see the future of machine learning in science and also outside of science?
I think there are two aspects. I'll say machine learning for medicine in the medical context, because then it's super broad otherwise. Like, so there are two things. One thing, like told you, there are problems in the clinical routine in terms of manual assessments or in terms of different types of variability. So you can mimic exactly what's being done in clinic. So you're exactly building models like for mitotic count, for example, or exactly. But then there is other part of machine learning or AI, they call it. Super sell out term. But it's similar, can be argued, but is to go beyond what is. So one is knowledge discovery and one is mimicking kind of. So and also there are multiple subfields as well. So I think the use cases for the mimicking is to improve the current clinical diagnostics, making assistive models for the doctors to have a better, more reproducible treatment plan and more faster and cost effective treatment planning. But then other aspect, which is super exciting, which comes where the science part comes in, is to learn if you can learn the new patterns which are not possible with the naked eye or the current limited biomarkers that we have keep on learning the new biomarkers, right? But whether we can increase this learning of new interactions, how the cells work, or how the human biology works, and given the amount of data we have, can we learn these patterns in the given data to have a new knowledge discovery?
But do we have access to those models, are they still like a black box?
No, right now people are working towards finding the new patterns and if they make sense. So that's where it's like, for example, if you can predict a gene. So we don't know a comprehensive list of genes. We don't know which are associated with the histomorphology or the morphology of a tissue. For example, let's say if I tell you have we have done human genome. We can sequence the whole genome now, right? So we have tons of genomic data for this particular tissue type, and you train a model to see what genes can be predicted. And let's say we can found a new set of genes which we never know can be associated. And we can reverse engineer and see see the molecular pathways these genes are type of, and we can maybe find, this is more of a causal learning, right? This gene is associated with this kind of changes in the histomorphology. So you are learning from the huge set of data a new information which was never known.
Are you guys already implementing this special transcriptomics, like a 10x genomics stuff?
We would love to do it, but we don't have money. I mean, no,
super expensive.
We have money, but it's
because exactly this is what you do. You do like tissue level, and they do exactly tissue level, but this is totally different slides. Actually, you might need two samples.
Yeah.
I don't know whether you can image at the same time, dissociate and sequence the same tissue.
Yeah.
If it is possible, then it would be amazing, of course.
Because
maybe you know spatial transcriptomics, right? So there's like on the tissue and exactly in the same spot, you have also the RNA expression.
Right, and then you have a spatial information.
Exactly. From H&E, you have the morphology and everything.
Yeah, but you don't have.
You have more like a different dimension,
Yeah, but I'm glad you mentioned it because, so for example, for the gene expression prediction, right now we are using bulk sequencing, which is like an average of all the expression. But we know intra-tumor or inter-tumor heterogeneity is a big problem.
Yes,
like for example, in grading a sample is not all. If like a tumor is grade 3, it's not grade 3 all over. It's only a portion or multiple parts are grade 3, and rest can be grade 1 or grade 2.
Exactly.
Whatever. So and these histopathology or the pathology slides or images are huge. They are gigapixel. So we generally break them into smaller patches. However, so for example, if you have a gene prediction, you are predicting a particular gene from these histopathology slides. Right now we are using bulk, which is a mean or average of the expression all over the tumor. But now we would like to see there is a concept of multiple instance learning, where we have a pathology image. We cannot give it directly to the model because of the computational limitations. Like GPU cannot hold these big images.
Exactly. But if you had special omics data previously showing you already where those expressions are getting weirder.
Exactly.
And then you can just use that pixels in your data, right?
So what we are doing, so we have smaller patches of these images. So you have like spatial information we are using or to predict the best average gene expression. So, but you have some predictive value for each of these spatial patches or the smaller images from this huge big pathology image. So what we would like to do is to see it, so it gives high attention to some areas and high attention or high expression value to some, and we would like to see if the model is actually learning the spatial distribution or spatial heterogeneity as well. And this can only be validated through spatial transcriptomics.
Yeah.
Then you have action, or you can even train a model with the spatial transcriptomics points. Let's say you have a huge, which is impossible, but you have a huge cohort with a huge number of images with the spatial transcriptomics. Then you have the ground truth, and you can train them, make the model learn the spatially. Oh, this morphology is more associated with high expression, or this with less expression.
Exactly.
So we are planning. This is part of our project, but we are still looking to expand this more subsequently, because I don't know if you know, it's 80 to 100,000 crowns for one slide or something.
I mean, the people who develop Vision, they're like in Scilab lab.
Exactly.
They are quite close collaborator with the 10x Genomics.
Yeah.
I think they are doing pretty much everything free since they are consultants.
Right. Okay.
I mean, nice to be in the KI part so you can collaborate, but exactly.
Yeah, we are definitely planning, but since most of our studies are like big cohort based, because
I mean, it's very cool. It's super exciting.
I mean, it's important to validate these models on different cohorts. I mean, because the biggest problem in machine learning modeling is
validation,
the robustness or the generalizability of the bias. So
maybe let's finish it with maybe your like a daily routine, like when you are in the lab, like how actually what you do, how it's going for you one single day.
And depends on what part of project you're in. So right now I started my new study, for example, where I'm trying to predict the survival or the recurrence of the breast cancer using only the using multiple types of histology images, like in these pathology images, you can stain them with different types of dye to get different types of biomarkers.
But you're in your desk, you're sitting in front of the computer.
Yeah, in that aspect, yes. So I can work remotely as well, which is a very nice thing. I can work from home.
So you have your laptop?
I have my laptop.
And you connect to the server, or how?
Yeah, we have. So since we are working with biology specimens or biology associated images, so they are sensitive data. Of course, we have pseudonymized. We cannot exactly translate back to the patient, but still we are working with the clinical data as well, and so I cannot have these images in my own laptop. Okay, so you don't know their names, sexes, and stuff. We don't know the name. We don't know the name. We know the person. You give your own names to them. You have a special images. Exactly. We call them my love. I mean, we have the names as the pathology A1, A2, A3. Yeah, or making more this hospital. But can you see the image? I can see the image. Okay, so it's allowed. You can see image. I mean, image, because looking at the tissue, you can never translate back to the patient.
Who knows? Maybe we can do it with AI as well.
No, you can.
I see your tissue, your breast tissue, and I can tell this is Abi.
No, I don't think so.
No?
Because
At least I can tell your age and sex, and maybe even your race.
Yeah, it's very hard. Race, you can say by the genetic makeup.
Yeah.
But not with the images only, you cannot do it.
Okay. Maybe in the future.
I mean, people in the pathologist, actually, some special case of tissue type, they put it on Twitter and discuss. For example, so you can never guess where this image is from, who is this. But if you just it's against the ethics to put the name of the patient. The name or anything which can relate with the patient should never be there on a public platform. So I can have the image in my laptop, but I cannot have the name, for example. But anyways, in my case, we have a server where we have and also these are huge images. You don't have enough memory or enough resources in your own laptop to run or to do anything with them. So we have a server, we have big machines with high number of GPUs, which are graphical processing units, to work with images. And then, yeah, I can remotely access them through server.
So how generally you have a smaller folder with a smaller size of images, and you test your model each time how it behaves until you create your algorithm?
Yeah, depending on the task, yeah, you can have a smaller sample. You can even use publicly available data or anything to start, like, okay, if this model training is working or not, or something like this, and then you go on and further. But most important part is you have a cohort, well-defined cohort, to for any study, and then the data splitting, like how this is my training set, this is my tuning set, how to tune. I will tune my model based on it, and the test set, internal test set, for example. So you have this set, and then this is where you develop your model. So this is a development set. I develop this model, then I test it, which I can use at one time. And once I have these model internally tested, I go and take that cohort from different country or different sites, and then as a external validation set for a particular problem.
And what is your most of the time is going for? Is it for writing a code, fixing the bugs, or talking with the colleagues, like
having a fika? I think it takes the most amount of fika is like a coffee with something sweet. No, but on a serious note, I think it's data pre-processing or managing the data takes a lot of time.
What do you mean, like managing the data?
I mean, you have slides come back from, so I don't know how to say. Let's say from one side you have thousands of 10,000 slides came up from this hospital, for example, and then you have to make sure to access these images, which are huge. Then you have to, these are macro images, so they are present at different resolution levels, and you extract them. Then you have there's a whole pipeline. Then you have this whole slide image, and then once you have this image at this resolution level, then you patch them or detect the tissue. What's the tissue area, because there's so much of noise. And then when you have only the tissue area, then you can patch them into smaller images, like tile. We call it tile. And then after tiling, you go on forward with, you have a model to detect the cancer tiles, only the cancer regions in this pathology image. So you have the cancer, and then and this is a whole process, especially getting the whole slide image, because one patient can have multiple. So you might have to find a primary slide, which is the main slide that contains the most amount of relevant information rather than.
Well, it's much more detailed than it.
Yeah, and so and also each step takes a lot of time to do, and
running the code is maybe the easiest.
I think the downstream task like prediction or running the code is slightly, and then you have to. That's where a bit of engineering comes in. Like you want to try different methods which are coming up, and you want to tweak blah blah. Basically, taking up public code base and see if it works on your code base or not, or something like this.
Would you like to advise anything to the students I mean, now, like, I think everyone is aware of that. The AI is getting a lot of attention, and I think they maybe should learn, of course, like programming and such.
I think the NVIDIA CEO said no one should learn programming anymore
Because of the ChatGPT.
because of ChatGPT, or it's getting super easier to code.
So what they need to learn?
No, but I disagree with that. You should learn. I think it's always a plus sign, even if you do wet lab, which is like, it's important to know the basic programming skills.
I think, yeah, because I don't know whether you read this, but they checked recently how many papers published had the text inside saying I am 2023 or 2022 ChatGPT. That's why I cannot say something about this year.
Yeah.
Or I'm just an AI, so I cannot be sure about this, blah, blah. So they actually forgot deleting even that part.
Right.
And so this is not like a learning and programming, right? This is just using without thinking and just copying and pasting.
Yeah, I completely agree with you. I mean, but what he really meant by is like, it's important to know what you want to do, because programming is just a tool to do what you want to do. Essentially, you want to it's important to know the statistical model that you want to train, or the machine learning model, the task. Why are you doing it, or why not we are doing it? That's why interdisciplinary knowledge is super important nowadays, especially between the computational and the biological side, for example.
So what would you suggest then?
So I would suggest like learn the basic programming language and statistics, which is super important. And then you have to figure out what problem that you really fascinate or you really like, because in the end, I mean, research is, you need two things: consistency and patience, which is like a basic human habit. But the only way you can go through it is if you really like the problem you are working on. There is no other way. So I would highly recommend to try different things which you find fascinating. If it's biology, physics, it can be anything. But take the programming side in hand, because every data you will generate, or everything you are doing is digitized. You will have a digital number for it, and if you want to be an independent analyst, or if you want to analyze independently, then it's always handy to come with. But I think explore the problems, like, oh, you want to work with how cell interacts, because most important thing is the lie they sell us in high school is we know everything. But as a researcher, I can say we hardly know anything.
Yeah.
So try to find a problem, explore different problem types, and be open to everything, not like specific things initially, and then once you find something fascinating, give it some time, and then if you like it more and more, then you will automatically go down the route of like the bachelor's, master's, or PhD, or the standard system that has been developed around research or academy.
Thank you, Abhi. It was a lovely interview.
Oh, thank you.
I learned a lot. I hope people gonna watch will learn a lot as well.
Yeah, I realized I used a lot of jargon. So please, I mean, I don't know, ask Jank, or ask if you can contact me. You can always contact me if you. But it was a pleasure. It was my pleasure. Honestly, thank you, Jank.
So thank you for watching us, and have fun.
Bye-bye.